Good afternoon. My name is Albert Brock-Gandel, and I'm presenting the poster on the NMR characterization of S. mutans protein antigen A. So S. mutans is a known etiologic agent for dental caries, and part of why S. mutans is such an effective agent is because of the strong biofilms which protect the bacteria from harm or treatment. These biofilms are reinforced by amyloid fibrils, which protect the bacteria. In this study, we aim to understand if antigen A and C123, which are two amyloidogenic proteins, are capable of interacting and working together to establish a biofilm. And we also identify an inhibitor that may be able to interrupt their interaction. We do this using NMR. Nuclear magnetic resonance allows scientists to quantify the electronic environment surrounding a nucleus. The first type of experiment we use is called waterlog-C, which allows us to see small molecules binding to a protein, which is helpful when screening for molecules that could be inhibitors. In waterlog-C, unbound molecules show a negative peak, while bound molecules show a positive peak. This can be seen in figures 1 and 2. The other type of NMR experiment we use is an HSQC, which allows us to see the electronic environment surrounding all the amines in a 15 nitrogen-enriched protein. In figure 3, you can see overlaid HSQC experiments where nitrogen-enriched C3 was titrated with natural abundance antigen A. You can see the two peaks that change the most during this titration in figures 4 and 5, which may represent a binding site, while the other residues that have changed might represent residues surrounding that binding site. So as we move forward with this study, we need to improve the assignment of antigen A. The trouble is that it's a very large protein and cannot be resolved adequately with HSQC experiments, so we're looking to use longer experiments that require the protein to be more stable. Additionally, we need to identify the binding site for C3 and EGCG, which is the inhibitor we used on the surface of antigen A, but hopefully this will paint a better picture of how exactly these proteins are interacting and how exactly amyloid inhibitors are able to interfere with their function together. Thank you for looking at my poster.